You may have heard of the term gateway drugs. Using gateway drugs such as nicotine or tobacco, especially during adolescence, has been said to lead to the use of harder, more addictive substances later in life. Adolescence is a time of rapid brain development, and the adolescent brain is uniquely sensitive to environmental influences such as substance use. The idea of a gateway drug is not novel, but the neural alterations causing the gateway effect is an ongoing investigation. For example, in adults, nicotine consumption suppresses the activation of immune cells called microglia, but in adolescents, this is not the case. In preclinical studies of adolescents, nicotine actually activates microglia, which leads to increased cocaine self-administration. In our lab, we're interested in the pathways through which substance use impacts brain function distinctly in adolescents and adults, and whether this investigation could yield a potential therapeutic target for substance use disorder interventions. Welcome to the Translational Addiction Podcast, where we decode the innovative research from the Lotvipor lab at UC Irvine dedicated to translational addiction studies and developing effective treatments. I'm your host, Michaela Samorton, and today with us we have a co-host. I'll let her introduce herself right now.
Hi, my name is Tanvi Garnani, and Michaela and I are both students at the Lotvipor lab at UCI. So to guide our discussion today, we're going to be dissecting the paper Neuroglia in Substance Use Disorders by Emily Castro, Shardad Lotvipor, and Francis Leslie on the role of glial cells in the development, maintenance, and treatment of substance use disorders. If you do want to check out the full article, which we highly encourage you to read, you can find it on the Lotvipor Lab website or in the credits of our podcast on YouTube. So the paper reviews preclinical and clinical studies regarding neuroglia and neuroinflammatory responses and how those contribute to substance use disorders. A substance use disorder is medically defined as the inability to control substance use, despite the adverse effects onto yourself or others. All drugs of abuse alter the function of our glial cells. Drugs of abuse can physically or chemically cause neuronal dysregulations and trigger an immune response mediated by our glial cells, which are the cells in our brain that aren't neurons. Glia are as abundant and potentially outnumber the neurons in our brain. The properties and functions of glia, including their exact number and role in homeostasis, is still being uncovered, but this paper reviews the current knowledge of glial-mediated mechanisms involved in substance use, and it evaluates the clinical evidence for targeting glia in therapeutic interventions. It also outlines the influence of the gut microbiome, which we discussed in our first episode, on glial cells after the exposure to drugs of abuse. What's particularly fascinating is the role that these glial cells play in substance use disorders might differ in adolescents versus in adults.
Before we begin our discussion on substance use disorders, let's dive deeper into the cells we are going to be exploring during this episode. As we mentioned, neuroglia are the non-neuronal cells in the central nervous system, which play a variety of roles, including maintaining proper neuronal function, launching the neuroimmune response, and contributing to synaptic plasticity and neurotransmitter modulation. So synaptic plasticity is the brain's ability to relatively strengthen or weaken synapses based on use and environmental influence, and it is particularly heightened during adolescence. Astrocytes and microglia are two types of neuroglia, and they are most relevant in drugs of abuse studies because of their central role in the neuroimmune response. As the principal immune regulators in our brain, astrocytes and microglia serve as our main defense against any foreign pathogens. Astrocytes mainly support metabolic and structural functions of neurons. They promote homeostasis in the brain's extracellular environment by releasing signaling molecules and clearing excess neurotransmitters out of the synaptic cleft. Astrocytes play a crucial role in modulating the permeability of the blood-brain barrier. The blood-brain barrier is a protective shield that separates the brain from the rest of the body's blood supply. Its primary job is to keep harmful pathogens out of the brain while allowing nutrients and oxygen into the brain. Microglia are mobile cells that contribute to the immune system by detecting pathogens, responding to external threats, and engulfing particles and debris in a process called phagocytosis. So phagocytosis is almost comparable to eating For example, when microglia come into contact with a pathogen or invasive debris, they will essentially eat or engulf this particle so that it cannot do harm to the brain. Microglia scour the brain for any external threat, and when they find it, they eat it in this process known as phagocytosis. When there's any external trauma to the brain, microglia are involved as well. Microglia survey their environment and initiate immune signals using cytokine and chemokine release after trauma. Cytokines are a signaling protein that act as messengers to regulate our inflammatory response, and chemokines are a specialized type of cytokine that chemically attract and guide our immune cells. Other immune cells can follow concentration gradients of these messengers to recruit help to damaged or infected cells. When our bodies are in an inflammatory state, reactive astrocytes and microglia are activated, leading to morphologic, transcriptional, and functional changes. The scope of glial functions in normal health and typical development is an emerging critical area of study for substance use disorders, which we will refer to as SUDs.
Drugs of abuse activate reward circuits in our brains, triggering positive emotions which then reinforce drug use. The ventral tegmental area and nucleus accumbens are areas of our brain involved in the dopamine-mediated reward pathway. The signaling and receptors associated with this pathway are studied as the mechanisms underlying substance abuse. Drugs of abuse activate toll-like receptors in our brain, which are proteins that detect pathogenic molecules. They're found in both microglia and astrocytes to activate our innate immune response and induce the release of pro-inflammatory cytokines. This means the activation of toll-like receptors leads to increased inflammation. During adolescence, our brain is maturing and its reward systems are developing. The introduction of drugs during this critical period may disrupt brain function in ways that differ from the effects we see in adults. This review evaluates the effects of drug use in glial-mediated activity and how the age of onset of drug use moderates this relationship. The paper discusses mechanisms of microglial alterations in treatments in the context of stimulants, alcohol, nicotine, and cannabis. But in today's podcast, we're going to limit our discussion to opioids and nicotine. So we'll start with the first drug class that the paper covers, opioids. Mechanisms of microglial alterations in opioid use are intricate and distinct, and they vary based on the age of drug use and the length of exposure to the drug. Neuroglia have opioid receptors, so intuitively they can be altered by the intake of opioids. The review assesses what gliosis patterns are induced by opioids, specifically focusing on the reward-related ventral tegmental area and nucleus accumbens. Gliosis is the scar formation in the central nervous system caused by the proliferation or the rapid increase of glial cells in a certain region. Gliosis is altered by the length of exposure to a drug, which will then in turn induce changes in behavior. The paper's authors want to see where microglia and astrocytes are most prominently expressed over acute and chronic morphine exposure. To achieve this, you can measure specific amounts of proteins or mRNA expression over different time points. For instance, in microglia, high expression of the protein CD11B in a particular area indicates increased microglial activity. Similarly, increased mRNA expression of the astrocytic protein, glial fibrillary acidic protein, or GFAP, indicates upregulation of astrocytes in a given area. Upregulated expression of both CD11B and GFAP are associated with inflammatory responses in the central nervous system. Whereas acute morphine exposure results in the increased mRNA expression of microglial CD11B and astrocytic GFAP in the nucleus accumbens, chronic morphine exposure results in increase in expression of both those proteins in the ventral tegmental area. So exposure to morphine over short time periods is associated with inflammation in the nucleus accumbens, but morphine exposure over long time periods is associated with inflammation in the ventral tegmental area. The upregulation of GFAP in the ventral tegmental area from chronic morphine exposure is abolished when treated with an opioid receptor antagonist, naltrexone. This allows us to conclude that it's necessary for opioids to bind to neuroglia in order to cause these regulatory changes.
Opioids induce pro-inflammatory neuroimmune signaling. For instance, a molecule involved in the inflammatory intracellular signaling pathways toll-like receptor 4, or TLR4, influences the reinforcing and motivational properties of morphine and heroin. We mentioned that drugs of abuse may differ in their impact on adults and adolescents. TLR4 is a receptor that illustrates these differences well. In adolescence, morphine binds to TLR4 and enhances TLR4 signaling and microglial activation in the nucleus accumbens. Preclinical studies show that adolescents, but not adults, exhibit reinstatement of conditioned place preference, or CPP, a measure of drug-seeking behavior. CPP involves administration of either saline or a drug such as morphine before placing an animal in a certain room. For example, a room paired with morphine may be a bright blue color, and another room paired with saline may have black and white lines in it. Preference for a certain room is measured by the amount of time spent in one room relative to the other. If animals spend more time in the drug-paired room, this indicates drug-seeking behavior. Reinstatement of CPP is a subsequent test where researchers first extinguish the initial place preference before reintroducing it. This is achieved by removing the drug until the animal no longer demonstrates preference to a certain room. However, CPP can be reignited by the presence of the drug of abuse, meaning if the researcher reintroduces morphine prior to entering the drug-paired room, the animal will again exhibit drug-seeking behavior. Reinstatement of CPP indicates a relapse and is paralleled by the upregulation of CD11B and microglial TLR4 expression. Reinstatement of CPP is not observed in adult subjects. In adults, opioid-induced reactive microgliosis in the ventral tegmental area reduces dopamine signaling and dopamine-dependent reward behavior. Microglia induces dysregulation in dopamine signaling, which contributes to impaired reward. Now that we have briefly identified the mechanisms of microglial alterations in opioid use and identified variations between adolescents and adults, we will explore one of the treatments used for opioids. Ibudilast acts to reduce reactive gliosis and is a TLR4 antagonist. It has also been indicated as a neuroprotective in Alzheimer's. It has been observed preclinically that the co-administration of morphine and ibudilast reduces glial activity, diminishes morphine-induced dopamine transmission within the nucleus accumbens, and decreases somatic signs of opioid withdrawal. Clinically, ibudilast decreases subjective ratings of severe withdrawal symptoms in people addicted to opioids. Ibudilast also reduces the positive subjective responses opioids have and reduces drug craving in humans. Other treatments for opioid addiction reviewed in the paper include pioglitazone, which is used to treat diabetes, propentylphylline, minocycline, and broad-spectrum antibiotics.
We will now continue our discussion with another drug class the paper covers, nicotine. Nicotine suppresses reactive microglia and promotes anti-inflammatory factors in the adult brain. It inhibits microglial proliferation and microgliosis, meaning it decreases scarring in the central nervous system. Nicotine also suppresses toll-like receptor 4 expression. Nicotine impacts the morphologic and functional properties of astrocytes depending on the presence of nicotinic acetylcholine receptors, which have the primary purpose of mediating neurotransmission between the central and peripheral nervous system. Nicotinic acetylcholine receptors allow ions like sodium and potassium to flow through a neuron, which causes an action potential when acetylcholine binds to them. Nicotine acts as an agonist in this context, meaning it binds to and activates nicotinic acetylcholine receptors.
Clinically, brain imaging studies in adults who actively smoke show decreased microglial reactivity compared to non-smokers. Preclinically, nicotine increases expression of microglial protein IPRA1 and induces pro-inflammatory microglial states in adolescents, which contradicts what happens in adults. The underlying mechanism for nicotine addiction and the so-called gateway effect is complex, but it involves the activation of the nucleus accumbens D2 receptors and bradykinin signaling cascades, which lead to the enhancement of subsequent cocaine self-administration. Nicotine, as well as other drugs of abuse, modulate dopamine release and bind receptors D1, D2, and D3. Microglia serve as a unique mediator in adolescence to reinforce the inflammatory response to nicotine, whereas in adulthood, the response is suppressed. In adults, microglia dampen D2 receptor activation after nicotine exposure, but in adolescence, it amplifies the D2 receptor activation, leading to hyperactivity and locomotor sensitivity. In addition to the overactivation of D2 receptors in adolescence, microglia and inflammatory responses are activated or enhanced in the nucleus accumbens, leading to susceptibility of cocaine addiction. So that was a quick survey of the glial dynamics in drug-induced behavior during adolescence and adulthood, reminding us that it's important to consider age when developing treatments for SUDs, or substance use disorders. A potentially viable treatment option that we'll be covering is N-acetylcysteine, which is a modified version of the amino acid cysteine, often prescribed medically. Clinically, it's been found to reduce nicotine self-administration, reduce cue-induced reinstatement, and decrease withdrawal. Chronic treatment with N-acetylcysteine induces long-lasting reduction in cue-induced nicotine-seeking behavior. In preclinical trials, N-acetylcysteine can decrease nicotine cravings, decrease smoking, and increase positive affect during withdrawal. Clinical randomized controlled trials were most successful when combined with psychotherapy and/or other nicotine cessation meds such as varenicline or bupropion. Taken together, adjunctive care with N-acetylcysteine might be optimal treatment for nicotine abuse disorder.
So, in our first episode, The Role of Gut Microbiome in Opioid Use, we discussed how the GI tract and central nervous system are connected through the gut-brain axis. The gut is even considered a kind of second brain because it processes chemical and electrical signals separately from the brain. We also highlighted the bidirectional communication pathways involving immune, hormonal, and neural connections between the gut and the brain through vagus nerve and neuroendocrine signaling. In the context of neuroglia, preclinical studies have demonstrated that germ-free mice have altered microglial morphology, altered density, immature phenotype, and a diminished inflammatory response. The gut likely influences neuroglia through two possible mechanisms: the vagus nerve or through the leaky gut, which involves translocation of gut bacteria into the circulatory system, which then triggers a systemic immune response. Leaky gut causes a release of more inflammatory cytokines and short-chain fatty acids. It also increases the permeability of the blood-brain barrier, which then influences neuroglia in the brain directly. The interconnectedness of the gut and glia make the gut microbiome a viable target for treatment for substance use disorders. If you're interested in learning more about the gut microbiome's role in the development and sustenance of opioid addiction, please refer to our first podcast, which covers each stage of addiction in detail in the context of the gut microbiome. So, if microbiota is altered, significant impacts on inflammatory markers in the circulatory system have been observed. Preclinically, alcohol and opioid use in tandem has been observed to lead impaired epithelial barriers in the intestine and leaky gut. Translocated microbiota trigger an immune response via host toll-like receptors releasing inflammatory cytokines. Disruptions may be rescued by short-chain fatty acid supplements. Preclinical studies have associated gut dysbiosis in morphine-dependent animals with the upregulation of neuroinflammatory genes and reactive astrocytes. Administration of probiotics such as Lactobacillus rhamnosus have been known to decrease cocaine-induced locomotion and microgliosis, as well as astrogliosis. The impacts of microbiome depletion on the brain and behavior as explored in animals reveals the potential of the microbiome as a potential therapeutic target for substance use disorders. Male mice with microbiome depletion by antibiotics lead to a decrease in morphine-conditioned place preference and morphine-induced locomotor sensitization. After a 7-day treatment with a high dose of morphine, changes in the nucleus accumbens expression are observed, including alterations to genes involved in gliogenesis. These behavioral and transcriptional changes are reversible by administration of short-chain fatty acids. In male mice, genetic depletion or antibiotic-induced depletion of the microbiome leads to enhanced nicotine-induced activation of dopaminergic neurons in the postventral tegmental area. This can be modified by the motivational effects of nicotine. This behavior was assessed using a self-administration model where mice could choose to receive nicotine directly to the ventral tegmental area.
Substance use disorders are a major health concern, especially within the adolescent population. The goal of this review is to explore the possible mechanisms for which glia may be used as a therapeutic target. The mechanisms behind drug and glial interactions is an ongoing investigation today. Of the treatments discussed in the review, ibudilast has been clinically indicated to reduce opioid withdrawal as well as decrease stimulant reward effects. N-acetylcysteine has been most successful clinically and preclinically. It is important to consider models of drug initiation and nature of dependence when developing therapeutic interventions. To measure the efficacy of treatments, preclinical and clinical studies measure time to relapse, cravings, and reducing use. Preventative measures may aim to decrease the development of tolerance or reduce reward feelings. It is critical to identify and understand age-related differences in drug-induced glia or glia-mediated treatments. Adolescence is a major developmental window during which exploratory drug use occurs, coinciding with the onset of sex differences. E-cigarettes have been a device simulating smoking tobacco that have been especially popular with adolescents, partially because of their design, marketing, and flavors. As we've explored in this paper, nicotine-induced glial dynamics differ between adolescence and adulthood, so the efficacy of glial-mediated treatments might need to be reevaluated within the adolescent population. Additionally, we must consider and explore the complex interplay between the gut microbiome, brain, and substance use, and consider how their connection could lend itself to possible interventions. We hope you enjoyed this review of preclinical evidence for interactions between the gut microbiome and substance use with associated glial alterations in our reward regions. Looking forward, future clinical studies may explore the benefits of short-chain fatty acid supplementation. If you're interested in learning more about the indications of neuroglia in substance use disorders, follow us on Instagram at Translational Addiction Pod for updates, and check out the infographics and papers in the Lotfipor Lab website linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipor Lab at UC Irvine, exploring how to understand addiction and develop effective treatment.